So our third talk and last talk before the break is Jehane Samaha, who is at the Elizabeth Wolkovich lab at the Arnold Arboretum of Harvard University. She's talking about variation in dormant bud size across species and range with implications for springtime phenology.
Should the lights go down?
Yes.
Hey everyone, I'm Jehane. I'm here to talk about bud set in temperate woody species and how the size of buds might impact their springtime phenology. So I'm here from the Wolkovich lab. We study temporal ecology and phenology, based at the Arnold Arboretum. And phenology, as many of you know, is the annual rhythm of plants or observable life cycle events such as springtime leaf out or fall color changes. And as many of you know, climate change and increasing temperatures may impact how plants behave and interact with other organisms and with the ecosystem. For example, by causing springtime phenology to occur earlier. So why is this important? One reason that the timing of springtime leaf out in forests is important is that the length of time that leaves are actually leafed out over the growing season directly affects how much carbon they can suck out of the atmosphere. So this is crucial for carbon cycling models. And also, these changes in phenology can cascade through an ecosystem, for example, impacting the ways that plants interact with their pollinators and then impacting reproduction of plants. Historically, phenology data has been very observational, so think about like Thoreau taking lots of observations around Walden Pond. But what our lab is doing is trying to figure out what exactly cues are triggering phenology in plants. So in order to create accurate models of climate change, we need to understand why springtime green-up is happening and what exactly is causing plants to leaf out at the time they do. And for this reason, our lab uses experimental methods as well as observational methods. So we have a couple of sites that we use to understand our plants' adaptation to climate regimes. And there's our, first of all, in Harvard Forest down here in Massachusetts, and also up an hour north of Montreal in St. Hippolyte, Canada. And starting next year, we'll be adding 2 more sites in the Second College Grant in New Hampshire, way up here in the northeast corner, and then also in the White Mountains. So we'll have 1 site at each latitude between here and Canada. So my professor posed the question to me early this winter, oh, what about buds? Why, when we're thinking about springtime phenology and what cues are driving it, we were like, well, buds are what comes immediately before springtime green-up. And the buds of temperate woody species display this huge amount of variation in size and shape, and they also leaf out at a huge range of times. So if you think about a dormant bud, it's essentially a packet of tissue that this plant is creating, saving over the winter and storing for later. But that's exposing the bud to a huge amount of risk over the winter and springtime. However, despite these costs, buds must have benefits because they exist. So plants are still making them. And we're wondering if the resources that trees spend on their buds pay off in the springtime and impact the timing of their bud break. So to answer this question, we need to gain this very specific understanding of what's actually going on in a bud and what is inside the dormant buds of different species. And this question of what's in a bud is a very old question. The best paper I found on this actually is from 1909 with Torrey Botanical Club, and they published a study of winter buds with reference to their growth and leaf content. So you can see here some old plates, beautiful drawings from that study, buds in cross-section. However, our goals are to develop some new metrics. We've got fancier tools now. We've got scanning electron microscope. So we're hoping to come to an understanding of bud traits through this framework of risks and investments and understand the choices that trees are making throughout the springtime. And thinking about assessing what, so on the left here here I've got a diagram from our lab of a continuum of investment across different life cycle stages of species. So, annuals with very cheap, fast tissues are investing a fairly low amount in their tissues, and adult trees with preformed buds are much more on the expensive tissue end of the spectrum. And if you think about it, the loss of lower quality, cheap tissue for a plant is a much smaller risk than if the same tissue was expensive and was killed off in a springtime frost event. So since preformed buds represent a fairly large upfront investment, we wanted to look at this investment across different species and understand the way that the risks of springtime frost events might be outweighed by the possible benefits that a tree could gain by leafing out earlier in the springtime and beating out canopy competition. All of that risk, though, could be mediated by protective structures in buds. That buds have these fantastic things like scales and pubescence going on that might moderate a lot of the risk they're exposed to. Bud set, in general terms, is the initiation of tissue the season before. So plants create their tissue at the end of the summer, or sometimes middle of the summer, and then save that throughout the winter. And dormant buds have increased freezing tolerance compared to other tissues. Then springtime growth begins with that preformed tissue. However, for indeterminate species like the fast-growing willow, the number of embryonic leaves that are set in the bud only represent a fraction of the total leaves that will grow from that location in the following season. And many species can exhibit both determinate and indeterminate growth, but they tend to lean one way or another. And I like to think about determinate buds as a basket full of eggs, that these are all the eggs you've got, those are all the nodes you're going to have for the season, and you probably better take care of them. You don't want to drop that basket full of eggs. Meanwhile, I think about indeterminate buds like a chicken. You might end up getting a lot of eggs out of that chicken. You don't really know, but it's sort of endless potential. So getting to some hypotheses that we're thinking about as we begin to analyze our data. First of all, I would predict that our determinant species, since they only have that one basket full of eggs, that they're experiencing greater risk, and that they might leaf out later than indeterminate species to protect those eggs from the springtime frost. Protective structures, I would predict, are mediating some of that risk. And likely we'll see some variation across our latitude and sites and the size of buds and also the springtime phenology that we're seeing. In terms of risks and opportunities, there are a couple ways it could go. It's possible that since some species invest more in their buds, that they're ahead of the game, they can leaf out earlier. On the other hand, it's possible that some species might invest more in their buds and not want to risk everything that they've set in there, so they'll leaf out later in the season. To measure these hypotheses, we've looked at how big buds are in terms of length and width and their dry mass, and also we'll be doing a lot of dissections under the scanning electron microscope and counting of embryonic leaves to understand how many structures are preformed in these buds and how big they are, and what's really being invested in terms of tissue development. And also comparing our species in terms of their determinancy. It's really cool how you can do this because there's no way of both counting the number of leaves in a bud and also letting that bud come to term, basically. So what you can do is you can count the leaf scars from the previous year's growth and compare that to the number of leaves in the bud and see if those numbers vaguely match up. We started out collecting dormant twigs out in our 2 field sites. And you can see here, this is our postdoc, Dan Flynn, on a lake in Canada in the middle of December. And we brought back a huge number of clippings. 1,600 of them went into growth chamber experiments. And that's where our phenology data came from for this experiment. So it's all relative data trying to understand which species from our list, from our sites, leaf out earlier or later than the others. The other half of the cuttings went into my bud measurement experiment, and then some of those were dried to get their dry mass, and some of them have been fixed in FAA for beautiful imaging and dissections. Here's a picture of the inside of the growth chamber experiments. So these experiments force different species to leaf out under different conditions of light and temperature to, again, our lab understanding what is really cueing these springtime phenology events. And then spent a lot of time in the fume hood fixing buds, so many buds. I was able to get incredible pictures under the SEM microscope. And so just a little show and tell right now because they're so pretty. Looking at some of the protective structures, this is a corallus cornuta bud, and you can see just how fuzzy and hairy that thing is. It's really beautiful. This is a leaf, an embryonic leaf from the inside of a corallus cornuta bud, and it's just the fuzziest thing in the world. It's great. This is the base of that little leaf where I pulled it away from the inside of the bud. And this is looking at a different species, Vegas agrindifolia, and you can see it's so much smoother. We've got a little bit of pubescence going on, but clearly a lot of variation between species. And their scales are just these smooth, hard. I'm sure you all have seen beech buds. Well, there's a picture back there. But the smooth cigar-shaped buds really, you can tell that they're smooth under this microscope. I will soon be dissecting these buds to image their meristems to really understand how many leaf primordia are preformed in the buds. This image down there is my first attempt at that with the Vegas bud I think broke. Here again is the comparison between the scales of corallus cornuta and Vegas. So in terms of measuring the buds, this is what so much of my time has been devoted to this winter. For the corallus species, you can see that there's a whole range of buds, but they correlate with each other, from the buds that are low on the stem to the buds that are at the tip of the stem. Some are longer, but they all fit one pattern. And then you can see Vegas is longer and narrower in general. To give you an idea of how much data we've collected, here's an overwhelming graph of all of our buds. We've got 6,500 buds from 27 different species that I measured this winter. And as we get the phenology data from the growth chambers all set to compare with this, we're looking for one of two patterns, or maybe both, or we'll see. But the first graph on the left over there, if we see that the bud size, if it's larger for early leaf out species, it might support the hypothesis that bigger buds help trees beat out early springtime canopy competition. The graph on the right, if we see this pattern with average bud size for each species compared with early leaf out, then species with smaller buds leafing out earlier might tell us that it's less risky to leaf out with smaller buds, so it could happen earlier in the springtime. So in general, in summary, we're thinking that bud traits might be a really important part of the story of how woody species make decisions about when to leaf out in the springtime, and that by linking these structural aspects of the buds to understanding the winter and springtime phenology, that'll help us to understand something about climate regimes in the future. And so stay tuned as we continue to look at this data and do dissections and explore it. Thank you. Thanks to everybody at the Arboretum and my lab, and people are wonderful.
You have some questions? Time for questions?
Yeah. This is really interesting work, by the way. Have you looked at how bud size varies by population within species?
Yeah, so that's the purpose of collecting them from two sites. We have some very preliminary results that might indicate that the buds from the same species in Canada are a little bit bigger than the buds in Massachusetts don't really have an explanation for that yet, but we're still like figuring out all the data to see if that's really true or not.
That's cool. I have a common garden of populus balsamifera growing right now, and when I go back, I'll have to check. Or I guess when the bud's set, I'll have to check.
Yeah, and that's a long-term project for our lab. Right now I'm starting seed from all these species for all our different sites, and that'll be growing at the Arboretum in the coming 10, 20 years.
I'm curious, you had on your chart of all the trees, you had some cracks in this, and I'm curious about like compound leaf trees or shrubs versus simple. Are you seeing differences? Because anecdotally it seems like the compound leaf stuff comes out later and drops earlier.
That's a really good point that it would be interesting to look at like the leaf shape. I'm also hoping to look at a little bit of the leaf packing within the buds. It's a more complicated thing to analyze, but there's so much out there on how leaves are packed and how that impacts the way that they can unfold.
Have you thought about considering whether the growth pattern is determinate or not, whether that would have implications for carbon storage?
That's interesting. I think that we're not doing much in terms of like, you know, CHN analysis of these buds, but that would be interesting to think about. So.
I was just curious about, were all of these samples collected in that same year? Because I wasn't sure if the temperature, the winter temperature before that summer of growth had an effect on what the, you know, the buds would be for the next year. You know, I'm not sure, you know, the different chilling requirements or the seasonality, and so I was just curious if
Sure.
data on that.
So all of these buds were collected within 2 weeks of each other, or really more within 1 week of each other. And we'll be repeating this experiment next year with all 4 of our sites. So that'll be interesting to see whether we see a lot of interannual variation between these buds.
So the things that you're calling determinate, are you saying that they don't have a capacity to neoform leaves?
So in general, I think that they have reduced capacity to flush out a second set of leaves from that node if they're determinate, yeah.
And that is that something that you're
Sorry?
trying to get at?
Yeah, so the way that we're getting at that is by trying to understand, so for example, in a willow bud, you might have, say, like 4 preformed leaves in the bud, and then on the previous year's growth, you'll see that there were 27 nodes of growth formed or something insane like that. Whereas for a determinate species, it might be like you had 8 preformed leaves and you get 9 the next species. So it's sort of all relative, but you can kind of see which way different species lean on that spectrum.
But are you going to have some way of determining whether your determinate species can neoform or if they have an early frost, or do you have the capacity to do that kind of experiment?
I think that we're going to have to assume that at some life stage, most of these species can produce indeterminate growth, especially for very young saplings. A lot of them have that potential. So it'll be sort of a general category, but we're not going to have much potential to say that this species is only ever determinate. Yeah. Ned?
So, because preformation is characteristic of all temperate woody plants, how much will you tie the period when those leaves are being formed, which is mid-late summer, to climatic conditions? So, you know, the constraint may not be on the spring, and it could potentially be really the summer before that determines whether you do 6 or 8 in your northern versus southern latitudes.
Yeah, I think a lot of it has to do with previous season growing conditions. And that's sort of a goal for me this season, is to be paying attention to all of our plants as we go through, to take some observations on when they're forming their buds and what the conditions are, because I'm really curious about that.
So I'm always thinking about, you know, if we're looking at costs and benefits, what is the currency or currencies that matter? And I know you said you're not going to be doing things like CHN, but I was just wondering if you could speculate what might be the most relevant currencies here. Do you think it's micronutrients, or do you actually think it could be like node number? That if you have to put some of your nodes at risk, that's the thing that drives risk versus reward decisions the most?
Yeah. So I guess what I'm gambling on with the metrics I'm collecting are that it is to do with something like node number. I am also saving all the buds that I've dried in case I want to do some CHN down the road. I think that in general, right now, what I've been hearing from colleagues at the Arboretum who do a lot of like sucrose measurements and all this type of sap flow is that all of that is really hard to calculate, and that it's probably not worth my time at this moment to try to figure out how much sap is flowing to the buds at different times. I think that probably carbon limitation is not what's happening because they've got huge stores in these trees that are not actually necessarily being stored in the buds for the winter that then come to the buds in the springtime they found.
All right, I think we can further discuss this, but I think we have to go off to our short break. We have a 15-minute break at the place where the registration was, and